So we want to welcome you today to the BDC Community Hours. The topic today is on linking phenotypic data and picture to genomic data files in 7 bridges. And we'll also have a presentation with the National Sleep Research Resource Center. So we're very excited. So just a few housekeeping things before we get started. Number one, you can see in the top corner the link, and it's also in chat, but we can repost that for you as well to follow today's slides. So feel free to use that. Otherwise, we always do at the start of each program our statement of conduct. Apologies if I'm looking away from screen. I have 2 monitors. So on one side, I have the information, and then I have my camera here, so don't mind. But let me go ahead and read this statement. So the BDC Consortium is dedicated to providing a harassment-free experience for everyone, regardless of gender, gender identity and expression, age, sexual orientation, disability, physical appearance, body size, race, or religion or lack thereof. We do not tolerate harassment of community members in any form, and sexual language and imagery is generally not appropriate for any venue, including meetings, presentations, or discussions. So that's our statement of conduct, and it is linked on the slides as well. When I have a moment, I can also post it in chat for you. In terms of today's agenda, we have our welcome that we're going through now and the housekeeping. And then we have 2 key presentations. So you have an introduction to the National Sleep Research Resource Center. And then we also have a demonstration and overview on how to connect genomic files to phenotypic data. And then, of course, we have time also for discussions and questions. So without further ado, I'm going to come off camera and off mic, but I'm going to continue to share screen for her. So Dr. Redline, are you ready?
I am ready. Thank you.
Fantastic.
Okay, and I apologize for staying off camera, but I'm having a finicky internet connection right now. But I did want to very much thank our hosts for having us today and everyone attending. We're very excited to be talking about sleep. Next slide. So what I will be doing over the next period of time is to give you a very brief overview of what is special about sleep and the excitement of having sleep be part of NHLBI data resources. And I'll provide an initial overview of NSRR and some of our approaches to data sets and metadata harmonization. And then my colleague, Dr. Sean Purcell, will actually share with us some of our approaches for developing and sharing data and sleep analysis tools. Next slide. To begin with, sleep is I think maybe for those who are on the call, you're already interested in sleep, but I wanted to emphasize that there is growing scientific as well as public health and clinical interest in the vast importance of sleep as a crucial pillar of health as it relates to nutrition, physical activity, and sleep management. And even the American Heart Association, as you could see by the bottom graph of Life's Essential 8, in just this last year, added sleep health along with 7 other components like diet, activity, blood pressure control as really critical to maintaining good cardiovascular health. Next slide. And not only is sleep integral for good health and actually interact with many of the phenotypes and disorders within biodata catalysts, but there's very exciting data about the importance of circadian biology and circadian rhythms. As you can see here is just a hypothetical biological rhythm that in health in blue, and maybe how it might change in red with disease or aging. And we now know that not only do we have there's a critical role of our central circadian clock in regulating multiple biological rhythms, but there's also many interactions with our central clock with many peripheral clocks that are in most of our tissues. So again, the really growing excitement of understanding both sleep and circadian biology as critical biological mechanisms and components of health. Next slide. But when we talk about sleep, and this is very relevant to those of us interested in sleep data, it's very important to note that sleep is not a single unit type concept, but sleep and circadian health can be described across multiple domains. And these may include sleep duration, sleep timing, rhythms, fragmentation, efficiency, depth, measures from brain EEG recordings on microarchitecture, macroarchitecture, clinical definitions of sleep disorders, and the patient-reported outcomes that describe daytime sleep sequelae. Next slide. And each of these domains may be measured by a variety and often quite different types of instruments, each of which really impact the types of data that we have available in our approach to visualizing, annotating, and analyzing the data. So for example, EEG-based metrics of sleep microarchitecture, sleep apnea often require a sleep, overnight polysomnography studies. Sleep-wake rhythms, on the other hand, may use, wrist-worn wearables or activity trackers. Issues about sleep, metabolic changes may use imaging data, and patient-reported outcomes will use various surveys. Next slide. And one of the really unique aspects of sleep data as we think about biodata catalyst and approaches of the National Sleep Research Resource is that sleep presents really maybe more granular and more temporally informative data than almost any other data domain. In fact, when we collect data overnight using polysomnography, we collect multiple, that's poly in the polysomnography, multiple physiologic signals over many hours of data collection. When we collect actigraphy or wearable data, we may collect data over days, weeks, or even months. And the collection of these multiple streams of data and over time provide many, many exciting data opportunities, but also data challenges. Typically, much of our research has taken these very rich data sources and collapsed them to very simple summary metrics. However, advances in our thinking and our toolbox now allows us to extract, very much more informative features. For example, the bottom of the slide, measures of hypoxic burden, or on the top of the right-hand side, quantitative measures of spectral EEG spectral analysis overnight. And these, again, provide opportunities for us to think about ways to share data and ways to utilize data. Next slide. And sleep data has some issues that are common to other data that are in BDC and other repositories, and some unique issues. As with other data sharing, data access, there are issues of ownership and regulatory type issues. But specifically for sleep, and although it's not unique to sleep, it is a feature we have to think about. Often, whether it's research data sets or clinical data sets, the data we collect is often heterogeneous. That is, it may be collected using a variety of equipment, what we call signal montages, or really approaches for how the data is filtered and displayed and sampled. It may then have different layers of human manual scoring attached to these. And the extent to which we understand those variations also varies. Sometimes they're very well annotated and other times poorly annotated, all of which give challenges to standardization, harmonization, the ability of any user to access these appropriately or combine the data across multiple data sets. We also have some special challenges that NSRR specifically is eager to address and has been working on, about the development of standardized terminologies and vocabularies and analysis tools, as well as the reaffirming needs to keep documentation consistent and clear. Next slide. So what it does the National Sleep Research Resource do? We actually started in 2013, and in the last 4 years, we've been supported by an NHLBI contract to specifically provide a community resource to overcome some of those challenges, as well as to take advantage of some of those opportunities of rich, temporally specific multi-channel data. And so it's a way to deposit and access sleep data, including those rich physiologic signals, including polysomnography and actigraphy and specialized sleep questionnaires. But it may include, according to the data set and the contributors' data restriction, a whole host of other data, including anthropometry, health behavior, physical activities, blood biomarkers, and so forth. And particularly one opportunity that integrating NSRR data and BDC data is the opportunity in many of the data sets to link our rich phenotype data with genetics data, and potentially in some cases even advanced imaging data. NSRR has also been recently ingesting data on sleep from animal experiments, as well as really complex circadian data. So those also are exciting areas. And as you'll also hear from Dr. Purcell, we're very committed to both develop and disseminate and document open source tools and support the sleep research community in terms of their engagement. Next slide. And we also have the NSRR platform itself also is committed to help users browse, search, and download the sleep data, and also help them with understanding differences between cohorts and accessing harmonized terms. Next slide. So when we think about a repository, you could think of it as a collection on the left of valuable, well-curated data sets, or if you go to the right, you could also think of a repository as really driving discovery and really supporting innovations in the research community. Next slide. And in this realm, our goal has always been to not only be a collection of data sets, but to add value and stimulate novel ways to think of analyzing sleep data. And we do that through improving over the original data we usually get of the documentations, providing search capabilities, doing certain amounts of harmonization and standardization for core terms and signals, providing metadata, improving, really adhering to fair and trust principles, developing and sharing open analysis tools, providing exemplar applications of our data, linking and integrating with other resources, including BDC, and building our community, including outreach and education. Next slide. So we have developed within the NSR itself mechanisms for fairly ready data access. And in fact, we have an online data use agreement that's fairly straightforward to complete, and that doesn't require an ERA Commons account. And because we have the data in NSR is not as sensitive as other places. We do not have genetics data, for example, and it's approved in general by our local data access committee. But we also have tried to encourage fairly straightforward and low burdensome sharing of data from the community with NSRR. And the process is outlined here, and for time's sake, I won't go into a lot of detail, but to say that we have a page on our website about data sharing, and it begins with really initiating a relationship with a potential contributor. We have some well-developed forms that contributors can access and complete to tell us about any limitations, scope of their data, describe the format of the data. We then meet with the contributors, we initiate a regulatory approvals, which does require a data use agreement just to share the data. and begin to understand really what those data sets look like. We then begin the sharing data process, and we often will we'll review initial samples of data and work with the contributors that way. We will ultimately, as I will show in several slides, we will then add that value to the data, reformat it, and annotate it and share what we've done with our contributors to make sure we were faithful to the purpose. And then continue to solicit feedback from the community on the clarity and the usefulness, and make quality improvement changes over time. Next slide. So where we are now, we have 26 data sets with another 8 to 10 data sets on the launching pad, just in really final stages of regulatory approvals and data modeling. These 26 data sets that we have now, though, include almost 50,000 individual overnight polysomnogram studies, about 30,000 that do have EEG studies, about 7,000 multi-day actigraphy studies. We've harmonized over 14,000 or annotated over 14,000 terms, including about 5,000 with detailed provenance information. We have about 5,000 data use agreements that have been executed, and we regularly share about 2 terabytes of data a week with the community. Next slide. Now, this is just a snapshot of our cohort matrix, and you could go to a number of tags to get an at-a-glance understanding of each of the cohorts. And in this particular slide, I'm showing you a data matrix, so you could see the 26 or so cohorts along the left axis, along that left column. And then if you go rightward, what you can see is the different folders that describe categories of variables that are organized conceptually, say anthropometry or medical history or sleep questionnaires. And the greens tell us which types of data can be found in each cohort. I do also, I know this is quite small, but in the circles on the far left-hand side are several of the cohorts that you will find in BDC. You'll find maybe some flat, simple sleep data currently in BDC, but the NSRR data, next slide, will actually provide further a deeper granular data on those physiologic signals, as well as those added variables and annotations, etc., that I described before. So our goal is to work as partners with BDC and really add sleep-specific domain data that can be linked to the other data in BDC. And after I speak, or after Dr. Purcell speaks today, you will hear an example of the first data set that's been migrated, Cleveland Family Study, CSF, and how you can link those data now with the physiologic signal data to genetics data, all in one platform. Now, next slide.
Dr. Redline, before we continue, we do have 2 questions in chat. Would you mind answering them now, or do you want to wait?
Yes, I could go ahead. The 26 data sets are all in NSRR. Okay. So you'll have to go to sleepdata.org. And the molecular omics and imaging data are not in NSRR. They would be in BDC for the selective data sets. So for example, we will be migrating data from Sleep Heart Health SHHS, which includes data from Framingham and Eric and CHS and Mesa, and there will be those omics data, et cetera, in BDC. So that's the opportunity. And I will let the picture team, the next question will address later when you see that demo, okay? Next slide.
Yes, yep, no problem. We can keep moving.
Okay. And this now goes into, again, just the approaches that we take to really make that metadata, the data that describe the data, useful as well as consistent. And we've created a number of forms that, like a metadata intake form, we ask our contributors to fill out, but we assist them with that. And and then we receive data, that can vary from REDCap forms to study protocols to the consent forms. And then when you get to the middle of the slide, then we as a team will work at developing that study level metadata, file level metadata, and variable level metadata. And the reason we do this is to produce a number of outputs that we hope is useful to the community. And we want to hear from you to find out how useful it is. And it includes some README files describing these data, as well as structured metadata and version-controlled standardized data dictionaries that we develop. In addition to the primary data dictionaries developed by the originating contributors, we also provide tools and data to look at summary statistics by various metrics, as well as we harmonize core terms, including terms that are in TopMed and terms that are particularly salient to our sleep community across data sets. So we actually do a lot of mapping and tagging. And again, you saw an example of the cohort matrix, some visualization tools. We also have some shiny apps to help people do various analyses and understand definitions and so forth. Next slide. And this is a little bit more about how we organize and do some of that detailed curation of metadata. So as I mentioned, all of our variables are sorted or organized in standardized folder structure to help with browsing. We work very hard on standardized terminology when often standardized terminology doesn't exist for sleep data, and we'll talk about that later. We also try to standardize the labeling of all the metadata, including details about references and scoring rules and scoring interpretations. And for selected variables, we apply tags to really help with either machine learning type queries or to help with some modern sleep questions. So for example, we have a compositional coding approach for all the hundreds of ways one could define an apnea-hypopnea index. So a machine could read that and know which AHI a given study utilized. And right now we're in the process of tagging various variables to map to a conceptual framework of sleep health using Dr. Bicey's Are You Sated framework. And then for variables where we can find inferentially equivalent terms, we do also harmonize these. And I need to give a big shout out at this moment for Ying Zhang, our data scientist, who has led this particular initiative and has done amazing work. Next slide. Yes, the answer is the meta standards. We are constantly reading up about community-wide meta standards, and they are, we try to be compatible with as many standards as we understand. That's an interesting question in terms of which standards people use, but again, the principles and the way to find these and utilize these should make these broadly interoperable. Now, as I mentioned, NSRR approaches to data processing are very much informed by the special types of sleep data that we take great interest in. And as you can see here, it includes actigraphy, the polysomnography surveys. Even for circadian, it will be including melatonin levels and other potential biomarkers. So again, a lot of our approach is to understand how these different data types differ, what their sources were. We often need to extract information, integrate them, and sometimes apply calibrations or transformations or other standardizations. Next slide. And here's an example. Actigraphy or wearable data, you might get it from a device like an actigraph, or you might get it from a device maybe potentially like a Fitbit. But we often use terms that have not been necessarily standardized consistently across devices and studies. So we begin with a conceptual model with getting input from our community members about what this device and this recording approach is intended to collect. So in this particular diagram, you see various metrics that identify specific data that are extracted from multiple day actigraphies. If you look at the big X's, recording start time, rest interval end time, rest interval start time, recording end time, you could see that you can identify 2 days, and we make it very clear in our data model what each of these, what's the difference, for example, between a recording start time versus a rest interval start time versus a sleep onset time, and so forth. And by identifying these clear intervals or time points, we're able to improve the consistency of the time, the sleep-wake periods that then are given annotations like wake after sleep onset or sleep efficiency or rest period or sleep duration. So this is one example of how we use a data model to then inform our maybe core actigraphy harmonized variables. Next slide. And as I said earlier, we do try to get the user very ready access to all the provenance of or the metadata related to each of the variables. And here is an example of an insomnia severity index, which is a standardized questionnaire. And you can see on the left side, you have the name of this particular variable, its label. There's a description of what it means, references. You could search for all variables in this data set that that are under this. You could see the mathematical calculation. You could find out if it's an integer, and you could find some synonyms or tags. And then in this particular data set, you could actually see the distribution of who had what level of score. Next slide. Now I'm going to turn the presentation over to my colleague, Dr. Purcell, who will now talk about the tools and briefly engagement. Thank you.
Hi, so thanks, Susan. And organizers, please feel free to interrupt me if you need time for the second component of this meeting. So we've spent some time working to complement our data resources in NSRR with appropriate software tools. And unfortunately, there's a real paucity, I think, of open shared tools in the field of sleep, as nicely documented by my colleague Tamar Sofa and her colleagues in this IMPRESS research note. So in the context of what some call a crisis of reproducibility across many biomedical research fields, we conducted a literature review of all sleep methods and algorithm papers published in a particular year. The vast majority of relevant reports did not publish either code, an example, or any data. And to be blunt, if a method can't readily be used by others, it's hard to see what value a paper describing that method really has. And of note, of 5 articles that supplied all of the above, 4 were published in journals that asked for code and/or data, pointing to a clear role for journal editors in this regard. Next slide. So in my own work and with the NSRR, we've developed an open source tool, Luna, mainly focused on sleep EEG, but have made some additional efforts to try to document and share this, to add tutorials, vignettes, and try to make the code base accessible via different means in order to share this as a resource alongside the NSRR data and facilitate others' attempts to reproduce our analyses. It's not always easy for small or moderate-sized research groups to do this, and this certainly is a work in progress in itself, but I firmly believe that something is usually better than nothing. Next slide. We've built an automated pipeline around this called NAP we use to process and harmonize PSGs, which we'll be instantiating in the context of BDC. There are some interesting decision points in how much to change the original data when doing this type of activity. We've ended up only fixing, hopefully, uncontroversial features of the data, such as labels and formats, that make the data easier to work with, but do not change the fundamental information. And beyond that, we attempt to, rather than attempt to fix things, we flag more subtle issues here. Next slide. We've also recently added this Moonbeam module where NSRR users can directly pull individual studies into the browser for interactive viewing and analysis. You can try this out yourself at the link remnonrem.net. But following from this kind of tighter coupling of code and data, This is really where we'd like, I think, to take a lot of this work moving forwards to support what I sometimes call a reference-based approach to the analysis of sleep data. Next slide. Back one. Oh. Correct, thank you. So that is, most analyses of PSG data are effectively self-contained in that new data are fed in, some derived metrics come out, but calculated only based on the current PSG. But following from other fields, including human genetics, I think there are at least two ways in which existing data can be used directly to contextualize and augment new data. So first, leveraging representative reference panels in whom the same set of metrics are being derived. One can imagine normalized, demographically adjusted centile scores for metrics, which can aid interpretation of new data. And second, if a reference database also contains other features that sufficiently track with the PSG features, those additional features can be statistically imputed via model-based prediction. So shown here, automated staging, where manual scoring is the feature present in the reference panel, is one form of this approach. But you can imagine many others, including prediction of disease state or risk, and as I've done with colleagues in the context of schizophrenia and other neurocognitive outcomes. And so we're starting to make more progress along these lines, for example, incorporating some age and sex-based normalizations of the sleep EEG and methods to predict so-called biological or brain age from the sleep EEG in pediatric samples. But I do think over time, these types of model-based analyses, tightly coupling code and data from NSRR and other sources, will really augment the value of a typical PSG or sleep EEG study. So watch this space. Next slide. So Susan said, we're very keen to build a community around these data and tools. So to that end, we have a blog where we give this kind of information, and we really solicit guest contributions as well. So if you have relevant work or you've used NSRR data and you think others would like to hear about it, please do contact us. We have a forum for questions and so on, and events and webinars such as this. And yeah, as Susan said, us hearing from you and getting feedback to tell us what's useful and what's not is really key. So please, either today or later, please feel free to do that. Last slide. So yeah, just acknowledgements. Obviously a large team of people here, and I think in the interest of time, I'll leave this up and pause, and we can move on. Thank you.
Thank you, Dr. Purcell and Dr. Redline. I'm going to stop sharing my screen so Emily can take over since her slides are next. But while we're doing that, are there any questions before we turn it over to Emily? I'll give her a moment too.
Alrighty, how does that look?
Looks great. Thank you.
Okay. Last call for any questions for now. Otherwise, we can hop into the next part of our agenda here. Okay, sounds great. So as I mentioned at the beginning, but in case we have a few folks joining later, my name is Emily Hughes. I work as a bioinformatics systems analyst at Harvard Medical School, specifically on the picture search platform on BDC. And so today I'll be talking about connecting genomic data files to phenotypic data. So before diving into the strategy or logistics on how to connect phenotypic data to genomic data files, I first wanted to introduce the two main tools on BDC that I'll be using today to demonstrate this. The first is the BDC Powered by Picture tool, and this platform empowers researchers to access data by facilitating approachable research for all skill levels. So it's essentially a search tool that enables data discovery and exploration, and researchers are able to search at the variable value and genomic variant level to understand what data exists. Researchers can then filter on those variables to apply inclusion or exclusion criteria to build a cohort. And once the criteria have been refined and the data selected, researchers can use Picture to export the data frame that's ready to go for research. This is all without opening any data files or mapping to any data dictionary, so really streamlines and jumpstarts the research lifecycle. The second tool we'll be using today is the BDC Powered by 7 Bridges platform. And Seven Bridges provides a secure workspace environment to do data analysis and collaborate. And analyses can be set up completely using a rich graphical user interface or through scripting via API or application programming interface calls. It provides private cloud-based Jupyter Lab, RStudio, and SAS Studio environments. And cloud compute and cloud storage are available on AWS or Google. And there are hundreds of accessible and hosted pipelines to get you started. So now that we have a general understanding of the tools, let's dive into the plan on how we're going to connect the data. So our main goal with the demo today is to connect the phenotypic data from the NSRR Cleveland Family Study, or CFS. For right now, this is the only NSRR study I think that is fully ingested by Biodata Catalyst and available, but we are looking to ingest more, as Dr. Redline had mentioned earlier, with those circled studies on the figure. And so we'll be connecting this phenotypic or sleep data from the CFS NSRR study to the TopMed genomic data from the same study, the Cleveland Family Study. And so I've provided the PHS numbers or the dbGaP study accession numbers there if you're interested in looking more into these. And so to do this, we will first use the BDC picture tool to search data, narrow down a cohort, and then add the variables that are needed to connect files. And I'll talk about this a little bit more later, but just to give you a general overview of where we're going here. So then once we've selected our cohort, we will export our data to the BDC 7 Bridges platform using code through the Picture API. And then finally, we will use the BDC 7 Bridges data browser tool to search and copy associated files over to our project. There are a few prerequisites to doing this process. So first, you must have authorization to access both studies or data sets that you wish to connect. So for example, if I wanted to link the phenotypic data from a parent study, call that study 1, to the genomic data of a TopMed study, so call that study 2, I must have authorization to access both study 1 and study 2. As a reminder, BDC uses the dbGaP authorization mechanism for data, so you will need to have a dbGaP data access request, an active data access request to get access through the BDC system. Additionally, to use the picture platform, you must have an ERA Commons account. Just checking the chat briefly here.
Emily, in case you missed it, there is a question in chat that's within a nested chat set. So
okay.
you already partially answered the first question, which was, can you easily find any sleep data in BDC? So if there's anything else you want to add to that, and then there's the second part.
Can you easily find any sleep data in BDC? Yes, I believe so. So if you're interested in phenotypes specifically, we do have those available in Picture, available for search. And then as far as the specific sleep files, so as Dr. Redline was mentioning, there are many different data types or data formats that NSRR collects and makes available. And so that is ingested by BDC, and you should be able to find those files on either the Gen 3 or the 7 Bridges platform.
And the second part was, what will make it easier to find and integrate sleep data in BDC?
I'm so glad you asked, and hopefully these next few slides cover it actually. So I'll go ahead and dive into that. All right, and it looks like Dr. Redline said TopMed has already harmonized a sleep duration phenotype, so that is also useful. Awesome. And so to accomplish this goal or this task, there are a few things we need to know when connecting data. And so the first is that the associated files often have some sort of identifier in the file name. And this is kind of the key, I like to think, to connecting these data files to the participants that you can get from Picture. And so to understand what identifier is available in the genomic data, I used the BDC powered by Gen 3 tool actually to view a list of files. And so that is what this screenshot here is. And so you can see I've boxed in red. In the list of genomic files, each file starts with a code NWD followed by 6 digits. And if you're familiar with TopMed data, you may recognize this as the TopMed sample ID. And so this is useful to know when creating your cohort and pictures, so we can be sure to export this information. And I'll be going into each of these steps a bit more in depth when I do the live demo as well. So another useful BDC tip for accomplishing this is that Picture has an add to export button. And so this means that instead of applying a filter on a variable, so that is this sort of action button here that looks like the filter icon, you can simply click the add to export button and it will add it as a variable to your growing data frame. And so this is helpful to use with the IDs that I mentioned on the last slide, and I'll be demonstrating this functionality later. So once we export the data, we are able to save the IDs that we have selected into a list using some simple code. As I mentioned, these IDs will be key to using in the tool later to find the data. And I did want to go over that the picture output formats participants per row and variables by column. So I've put sort of an example of this, just some fake data with variable 1 and variable 2 to give you an idea of what this picture output could look like. And then finally, the final tip or strategy that we're going to use to accomplish this is the data browser feature on 7Bridges. And so this is an extremely handy tool that we will use to search our terms of interest. So using those IDs to locate associated files and copy them over into our project. And so once those files are available in our project, then you're good to get going on adding workflows and starting your analysis. You have all of your data in one place. So I will go ahead and hop into the live demo here. So let's see. I will go ahead and start with the Picture platform. And so here I have logged into Picture, and to make sure that I have the correct data access permissions, I have logged in with my developer account. But as I mentioned earlier, you will need to use your ERA Commons credentials to access Picture. And so with my developer account, you can see I have access to all of the studies available in Picture, but these numbers will likely be different based on your personal authorization to access data. So I'm happy to talk more about some of the other features on Picture, but for right now, I'm going to focus on this authorized access. So authorized access allows you to search and query from all data sets that you have been authorized to access through dbGaP. And so here to start with a research example, after speaking with Dr. Redline, she had suggested that perhaps looking into apnea-hypopnea index would be interesting with age and sex. And so I did a quick search yesterday on what this meant. So, I'm glad there are sleep researchers on the call to correct me if I'm wrong. But from my understanding, the apnea and hypopnea index is the combined average apneas and hypopneas that occur per hour of sleep. And it looks like this is actually separated into 3 general categories: mild, moderate, and severe, where the mild is 5 to 15 events per hour, moderate is 15 to 30, and severe is greater than 30. So we will be using this as our phenotype of interest along with sex and age, and also get our cohort built so we can export to 7Bridges. So any sleep researchers on the call, feel free to chime in if I explained that incorrectly or if more details would be helpful. But I will go ahead and start. Let's say sex first as our first phenotype or term of interest. So when I search for sex, this is returning all of the variables that match the search term of sex across all studies that I'm authorized to access. So this could be in the variable name, the variable description, the variable values, the study name. Any part of the study that has a match for sex is returned as a search result. So you can see that there are over 300 variables that match, but I'm really interested in the NSRR data, right? So I can go over here to the filter search results by study tags and find the NSRR CFS study tag, which I think is right here. So you can see that this tag exists here, and I can click the plus sign. And now my search results are narrowed down, and I can see that all of the search results here are only from the NSRR CFS study. So this is useful to narrow down search results of common terms. So looking through here, I can see that participant sex seems like a good fit for my sex phenotype of interest. And so I will click on the filter button here in the actions column. So by clicking on that, I can see that this sex or participant sex variable is a categorical variable. So as expected, there are values for male and female. I can either check the box next to each or click the select all button up here in the corner. So if I click select all, both values are put over into the selected values column. So I'll select both, and when I click add filter to query, you can see that a few things have changed in the results panel over here. So first of all, my total number of participants has been updated to reflect my query criteria. So this means that there are 710 males and females in the NSRR CFS study. You can also see that this variable has been added to the added filters box down here. So this kind of gives you an idea of what you've added so far, how you've filtered down your cohort thus far. So next I can search for that. Oh, yes.
May I ask a question that are related to what you are doing right now? So what if I don't know the NSRR is the sleep? I want to search for any sleep related data. Can you do a search and find out which cohort that has sleep data?
Yeah, so I can do that at the end here. I'll go ahead and finish my the demo of this cohort specifically, and then I can go back and do that. Does that sound good?
Thanks. Yes.
Okay, awesome. Sounds good. All right, so just moving forward here, we will search for the apnea-hypopnea related variable. So this is the apnea-hypopnea index. And so as I was searching here, Dr. Redline provided some insight that perhaps this sort of variable could be of interest. So I can go ahead and again click the filter icon here. And so we can see that this looks a little bit different because this variable is continuous, right? So there aren't distinct categories, instead it is numeric. And so let's say I'm a researcher interested in moderate and severe apnea-hypopnea indices. So I can do greater than 15, and I will go ahead and leave the max blank, so that way the default will be the maximum that's in the data itself. So when I click add filter to query, again, we can see that my total number of participants has updated. So now there are 173 participants that have a apnea-hypopnea index greater than 15 and are male or female. And the final one that I'll do here is just an age variable. So if I search age, we can see that there are quite a few variables that are returned, right? So there are 390 variables that are returned for age. Now, let's say that I'm interested in age categories. So I can actually add, using the variable tags, the categorical variable tag here. So let me click the plus sign next to this. And so now there are 59 variables that match my search, but I still want to find participant age somewhere, right? So I can see that age diagnosed, I'm not necessarily interested in that. I just want the participant age in general. So going over here, I can see that another variable tag is the tag diagnosed. So let me actually exclude all variables that have the term diagnosed in it, because that's not really what I'm interested in right now. So by adding that, I can then take a look at my list here and see, okay, participants age category. That's exactly the variable that I'm interested in or that I'm looking for. So that's just to demonstrate how these tags can really help with your data discovery and data exploration.
So Emily, two things in chat. One's a comment from Dr. Redline and the other one's a question. Is it okay to pose those right now?
Sure, yeah.
Okay, so Dr. Edline said that if you see the long list from earlier, when you first did the search for age, that you can see that for sleep data, NSRR data sets may have very detailed information on variables such as AHI, which may be defined multiple ways. So just supporting what you're doing.
Awesome.
And then we have a question from one of our participants today, or attendees. Can we use command, for example, our language, to repeat the all operations that you just did on screen just a little bit ago?
Yes, that's a great question. So yes, there are two ways to access picture. One is through the user interface, which is what I'm showing right now, and the other is through code. So we have availability in both Python and R. So if you see any searches or any filters that I'm adding on screen today, there is an equivalent in either Python or R as well. So if you're more comfortable or more experienced with code, that is definitely an option.
For the access to the studies on BDC Picture, is it through dbGaP for authorized access?
Yep, so that's exactly right. Like I said earlier, the Biodata Catalyst system uses the dbGaP authorization mechanism to determine what researchers are authorized to access or not. So in order to apply for access, you will have to go through the dbGaP data access request process. So once you're approved from dbGaP, you'll be able to log into BDC, you'll be able to log into Picture, and you'll see the available studies pop up. Great questions.
You can keep going now. Thank you.
All right, so I will just quickly, add a few things here because I just noticed the time. So the last thing that we are interested in, so as I mentioned earlier, we do have the TopMed sample ID, and this is sort of the key. Sorry, this is finicky. This is sort of the key to linking the phenotypic data to the genomic data files. So if I go into Picture, I've cleared all of the tags, so I'm searching across everything now, and I search sample ID. This search usually takes a little bit because a lot of hits are returned, but right now I'm searching for the TopMed studies sample ID, and I'll be able to add this to export as an additional column in my data frame. So here you can see there are a lot of sample ID, over 29,000 variables that are returned. But if I scroll down here to the CFS study, so that's the 954. I can find the variables only for this. And so you can see that this is the CFS TopMed study, and I'm going to add to export this sample ID. And so by clicking this button, I add it to the export. And so then you'll be able to package the data using the Select and Package Data tool, and copy your token and your query ID and use the Picture API then to export data to 7Bridges. So let me just quickly showcase the data browser tool in 7Bridges to kind of show you how this would work. All right, so I've created a project in 7Bridges, and this is what it looks like. And so I've actually already done the export with a new data studio. So this data studio is called NSRR Data Retrieval. And so you can see based on, I just saved this code that I created as a PDF so you could see, but you can see that I'm able to quickly connect to the Picture instance. Don't worry about all of this output here. It's just saying, hey, you were able to connect. And then I paste my query ID that was generated in the Select and Package Data tool, and I'm able to export the data. So as each participant is a row and each variable is a column. And now what I can do is actually use the sample ID column to create the names of the files that I want to search for. So as I had mentioned earlier, if we go back here, we can see that the data name or the file, genomic file name is the sample ID followed by this text here. So I've essentially just created a list of those. And I saved that to this output here. So let me just copy all of these. So I have them on my clipboard. And then to use the data browser tool, you simply go to the data browser, the data tab, and click data browser. And so here we want to search for CFS because that is the Cleveland family study is what we're interested in. And we want to search the TopMed files. So I will click the TopMed study and click explore. And again, I want the files associated with it. So I can click file. And then finally, I can click this search by ID button up here in the top right corner. And so here I'm able to paste all of the files that I've created based on that sample ID and click next. And you can see that there are 100 file hits that are returned, which is expected. And so if I search this, I can see that all of the files of interest, so all of the files based on the cohort I've built have been selected. And now I can copy files to project. So by clicking on this and selecting the project of interest, I can copy 100 files to my project here. So I have just copied things over. So let me go back to my project just to make sure. So by going to the Files tab, I can now see that all of my genomic files based on the cohort I've created in Picture are available here. And so now you're ready to get going on your workflows, on your analyses, and have it connected to the phenotypic data based on the sample ID. All right, so I know we're right at time, but I'm happy to take any questions. I think that pretty much covers everything that I intended to cover.
I also have the poll running since we're pretty close to time in case people have to jump right away.
All right, so I see some chats here.
Some of those chats are just me and Dave just helping out for a closeout in case people had to leave like 5 minutes early.
Yep.
Emily, while you're waiting, could you be able to search any other TopMed cohorts that has sleep data? Can you do that?
Sure. So if we go back to the picture tool here, and I'm just going to click reset just to start with a blank slate here. And perhaps I guess I can just start with sleep. That seems like a pretty solid search term for this sort of query. So by clicking search, we can see. Sorry, I'm managing my Zoom here as well. It looks like there are some studies that have some additional sleep data. I'm not sure exactly what sleep data you would be looking for, but there are sleep phenotypes or sleep questions that have been asked of participants in other studies.
The number next to the cohort, are those the number of sample or number of variable?
Are you talking about these
Yes,
numbers
yes,
over here?
right there.
Yep, so that is the number of variables that match your search term.
Variable, okay.
Yep.
Okay. What about imaging? Can you search imaging, see which cohort has imaging?
Yeah, so that is functionality that we're actually working on building right now. So it's not available presently, but as BDC works to ingest more imaging data, we will be expanding the picture search mechanism to include searching imaging files available.
What about hypertension variable?
Hypertension, yeah, we have some of those, I believe.
Good,
So,
good.
yep, you can see. So it may be a little unclear at first of why these are related, but there's likely a variable value that's associated with those. But yep, you can see all of these hypertension related variables.
Okay. What about heart failure?
Yep, I think we should have those as well.
Okay, that's good. That's great. Thanks.
And
Yeah,
I will say,
oh, sorry,
oh,
Emily, go ahead and then I'll add something.
Was just going to say the open access tool, which I didn't have much time to dive into. So if you have an ERA Commons account and are not authorized to access any data quite yet, but want to figure out what data sets are available, what variables are available, and what to request access to, you can use this open access feature to do these searches and kind of get a feel for what data exists prior to having access. So that could be a good tool as well.
That's
and-
more important. Thank you for that.
Yeah.
Yeah, and I was just going to mention too that heart studies are our biggest percentage of data on BDC as well. So you can find a lot of heart study details, I think. Safe to say, Emily?
Yep.
And then we do have at least 2 questions in chat. So one person asked, by genomic data, we mean only variant data. Is that correct?
I believe so, yes. So you're able to search by gene variant on picture, but the whole genome sequencing information is available through TopMed, I believe.
Yeah, and Dave mentioned sequence reads as well and variants
Yep.
too in chat. Just checking if there was anything else to add. We do have several questions coming in. So next question is, which Python or R API is used to extract data from picture?
Yep, it is the picture API. So I can definitely send some so we actually, let me go over to the picture project, I believe. So on 7 Bridges, if you want to see an example of how to use the picture API to export data, you can go to the public resources and click projects. And you'll see here, there's a picture API tile. And so if you go in here, you're actually able to see in the Data Studio section, you can see there's an RStudio example or Jupyter Lab. I think since this researcher is interested in R, we'll click on the RStudio. And so, oh, here, wait, maybe not. Yes, so here you go. So you can see we have like a 101 example notebook or example R markdown file that teaches you and provides you with code that you can run step by step to actually use the picture API. So here, maybe I'll just throw this in the chat now. It looks like there's also a question about, is there a list of variables? If you're interested in the full list of variables, you can search the study name or the study PHS number on picture, and that should return all of the variables. Another good place could be to go to, so we don't have the data dictionary files available on picture because we want to incorporate this search mechanism. But I do think that perhaps 7 Bridges might have a tool, but I don't want to speak on this because I'm not an expert in 7 Bridges.
Emily, a quick follow-up question. So on those numbers, you said next to cohort, you said it's a variable. Do you have a number of case or patients from that cohort that has that variable?
No, you will have to go in and do a filter on these variables to determine that, right? So if you're interested in, the ORCID study is a COVID-19 hydroxychloroquine clinical trial. And so if you're interested in how many patients have congestive heart failure, you can, select these, and then the total number of participants will be displayed over here based on that.
Okay, that's not available through open access, right?
That is available. I'm on open access right now.
You're
Yep,
on
so
open access.
it is.
Okay, good.
Yep.
Talking about sleep studies and linking it with heart study information with Matthew Goodman, who's from one of the current TopMed fellows. So I think that'll be really timely to continue the conversation from today.
Yeah, I'm glad that you mentioned that. So the link between the phenotype and the genotype is only by the NRSS or other sleep cohorts that is in the TopMed data sets.
Yeah, so if I understood your question correctly, you'll be able to link the phenotypic data and the genomic data of any related studies on BDC, right? So like with parent and TopMed studies, or this NSRR data set and the associated parent or top med study with that. Does that
Yeah, I
make
thought
sense?
if you want to link the same patient who has phenotype data and the genotype data. And some of the patient in the TOMAT cohort may not be in NSRR cohort. So you're not linking those, right?
And what permissions are allowed to cross-reference between studies, right? So what happened to the study before it even got ingested into BDC, partly. But Emily, you might have additional thoughts too on that.
No, yeah, I think that clears
Yeah,
it.
I think you have to link the same patient of their phenotype with their genotype. Otherwise, it's not meaningful.
And
Right.
Dr. Oh, go ahead. Yeah, I was gonna say Dr. Redline just posted in chat about it too. Is the difference is that the signal phenotypes so far are only in NSRR, but they are being migrated as in the Cleveland family study that we talked about today. So yes, we are going to be over time adding more things that link better, but this is the start.
Okay, thanks.
Yeah, and then we do have a couple questions that were asked about the recordings. I just wanted to say them out loud, that the recording should be posted within about 24 hours into the forum post that's referenced on the slides. And then there was a question from somebody asking, they watched some videos on YouTube and mentioned that both Gen 3 and Picture can be used to select phenotypic data. So what is the difference between the two?
Yeah, that's a great question. So the way I think about it is that Picture is much more variable focused. So you will search and filter at the variable level, whereas Gen 3 is a great tool for researchers that are trying to find files. So if you're interested in finding associated files with this or files associated with a study, Gen 3 allows you to easily browse those and find those and export those to an analysis workspace. I do believe that Gen 3 does have some phenotypic data filtering available, but Picture, I think we actually ingest the study and actually display the variable levels. So if you're looking to filter on variables specifically or discover data at the variable level, I think Picture, that's where the strengths in Picture lies. So yeah, it generally Gen 3 files and Picture variables.
Yeah, and I think that's it for all of the questions. I did want to highlight, Emily, if you just go back to your slides that you have open, or the slides from today, we didn't get to it because of sake of time, but there is a little video GIF on slide 42 for everybody that just showcases everything that Emily talked about in terms of how to do the linking. So just wanted to make sure that those who are still on the call are aware about that. So you do have something to remind you of what to do if you forget after today.